Hello and welcome to this virtual undergraduate research symposium. My name is Emily Nelson and I am here representing the Biomedical Engineering Department. The title of my project is Osteoconductive Electrospun PCL beta-TCP nanofiber scaffolds. Through this research, I considered nanofiber scaffolds with the potential to support bone growth and regeneration. To carry out this work, I used electrospinning to obtain a polymer nanofiber scaffold and mouse-derived osteoblast precursor, or MC3T3 cells, for in vitro analysis. For this project, I hypothesized that the scaffolds containing beta-TCP will improve osteoconductivity of the scaffolds and support growth of the osteoprecursor cells. Experiment setup consisted of using Polycaprolactone, or PCL, nanofibers with beta-TCP that I electrospun as a substrate for the cells. Down here to the left, you can see an image of the fibers that I spun with a honeycomb structure which we previously found is optimal for cell growth. A little further to the right on the top here, you can see an EDX composition analysis showing that the TCP is incorporated within the fibers. Mouse-derived osteoblast precursor, or MC3T3 cells, were used to determine the cell growth over 24, 48, and 72-hour time points. Using cell titer blue viability assay, I determined the viability of the MC3T3 cells on the test scaffolds, a control PCL scaffold without beta-TCP, and a standard tissue culture plate. Data indicated that the number of viable cells on the PCL beta-TCP scaffold were at levels comparable to the PCL control and the standard tissue culture plate. If you look at the chart down below, you can see that there's expected decrease of growth at 48 hours and then recovery at 72 hours. To determine significant difference in cell growth of these scaffolds, a systematic study of varying concentrations of beta-TCP and long-term cell studies are needed. Future work with these scaffolds includes immunostaining, Western blotting to identify specific biomarkers affected by the scaffolds. Additional experiments are also needed to identify the concentration of beta-TCP needed to affect the cells. In conclusion, I would like to thank the Pavlis Honors College and Portage Health Foundation Undergraduate Research Internship Program for their support in this work. Thank you.